Hello, everyone. This is Tarek again. This is our last lecture of Block 2. We'll continue our discussion of DNA damage and repair. So let's get started. All right, so in this lecture, we will follow up on what we discussed in the last lecture about the different pathways that are dedicated to the repair of DNA damage. And this one will, as I mentioned in the last lecture, will focus primarily on the repair of DNA double-strand breaks. All right, so here's a brief outline of our today's lecture. We're gonna briefly go over what we've discussed last time. We will discuss the different cellular responses to double-strand breaks, primarily focusing on the concept of the DNA damage and repair pathway. We will focus on checkpoint activation, focusing on three important key players in this response: ATM, ATR, and the tumor suppressor P53. We will then go over the major pathways that are dedicated to the repair of double-strand breaks in mammalian cells primarily. That's going to be the focus of our discussion. We will discuss two major pathways, non-homologous end joining repair, as well as homologous recombination repair, which is also involved in the repair of interstrand cross-links. And then at the very end, if we do have some time, we'll see how that goes. We will see how scientists in the field are able to detect and quantitate the repair of these lesions. All right, so this is our favorite slide from last time. And what we've done in the first lecture is focusing on these different damaging agents that primarily affect the structure of DNA, different damages that are influenced by different DNA damaging agents and are repaired via multiple pathways that have some common themes in them, which is the ability to remove the damage, well, first to detect the damage, then to remove it, and then ultimately to seal the gap and restore the sequences to limit the amount of mutations that potentially are affected. Today, we'll focus on the repair of double-strand breaks, and again, it's important to realize that double-strand breaks are probably the most lethal of all kinds of damages that can affect DNA. They are the most serious form of damage that can affect DNA, as we just said. And most of the lethality actually comes from the aberrant, or what we call inaccurate end joining between different breaks that can happen throughout the genome. These obviously can result into chromosomal rearrangements, genomic instability, oncogenic transformation, and can ultimately lead to cell death. Sometimes these breaks can be joined inappropriately, but do not necessarily cause lethality. For example, here is one such example where the repair of these lesions happen between different chromosomes in a sort of symmetric fashion where one segment of one chromosome gets joined with another segment of a different chromosome. And so that's called symmetric translocation. You can see that with the exception of the rearrangement of the chromosome, the structure of the entire chromosomes are intact, and the different genomic composition of these chromosomes are still intact, but in different chromosomal arrangements. Of course, sometimes, depending on where the translocation happened, this could have some serious consequences, but they're not necessarily lethal. So the focus here then is to look at some of the inappropriate ligation step between different breaks that can be happening when you have extensive amount of these DNA double-strand breaks in the cell. And these could be inflicted by many different forms of agent, as we mentioned. Radiotherapy as well as radiomimetic compounds can induce many cytotoxic double-strand breaks in the genome that are likely to be cytotoxic to cancer cells, of course, depending on the dose and the frequency at which ionizing radiation is being inflicted. And this is how radiotherapy is very effective. In fact, radiotherapy is still the most effective single agent that's dedicated to the killing of cancer cells. Other agents such as cisplatin or platinum compounds in general, mitomycin C, for example, these are bifunctional alkylating agents that causes DNA cross-links between either different nitrogenous bases on the same strand. These will be considered as intra-strand cross-links, or more seriously, cross-links between the two different strands of the helix, causing interstrand cross-links. And when these get repaired, they often involve the formation of double-strand breaks, as we will see. But if these happen at a very high frequency, then these double-strand breaks cannot be repaired and ultimately kill the cells. And this is why platinum, for example, is still probably the most commonly used chemotherapeutic for cancer patients. Additional agents like topoisomerase inhibitors, these are also part of the chemotherapeutic regimens that are commonly used in cancer patients, and these causes double-strand breaks through some other mechanisms that primarily involve problem with replication forks, as well as the formation of replication fork blockers that ultimately result into double-strand breaks as well. There are other agents that are a little bit less associated with double-strand break induction. These are typically our replication inhibitors, acetylcholine and hydroxyurea. Hydroxyurea used to be considered for chemotherapy in the past, but no longer used at any noticeable frequency. Acetylcholine is another agent, and both of these guys what will do is they sort of deplete, either prevent the polymerase, the normal polymerase from driving DNA replication, so they're an inhibitor of DNA replication, or hydroxyurea, which is a drug that is used to deplete the cells of the nucleotide pool, and by doing so, cells have problem finishing DNA replication, and they result into stalled replication forks and ultimately collapse and forming double-strand breaks. But they're used still very extensively in biomedical research for different purposes. So clearly then, it's important to understand how double-strand breaks are repaired in the cells, because if they're not, then that can result in genomic instability, cancer development, as well as sensitivity to different chemo and radiotherapies. And that's why it's really important for us to have a good grasp on how the cells repair these lesions. All right, so let's see how the cells respond to double-strand breaks. There are two key steps in that process. The very first one is the rapid activation of cell cycle checkpoints. This is really critical because what that does is that it gives the cells some time, depending on where they are in the cell cycle, which you've covered already in the cell cycle lecture. It gives the cells some time to repair the damage before they continue dividing. I mean, this is really critical for dividing cells. It's not as much important in quiescent cells, where they are by definition arresting. And so they have all the time there is to repair these lesions. But in dividing cells, this is a really critical step. You need to arrest the cells wherever they are. If they are in G1, you want to keep them in G1 before they commit to entering S phase. If they are in S phase, you want to stop them right there before they duplicate the genomic material when they have already severed genome. Or if they already have finished replication and they're now in G2, you want to make sure that they are arrested so that they don't pass on damaged DNA to daughter cells. The second key step is the recruitment of DNA repair factors that will come onto these packaged genome and allow for the DNA double-strand break repair protein to be loaded in order to restore the integrity of the genome. You've heard from the chromatin lecture that remodeling is an important step in many different physiological responses, and one of them is in response to DNA damage, because you obviously want to consider that the DNA normally is not naked DNA, it's actually covered by chromatin, and that chromatin may impede or facilitate the recruitment of different proteins. And so for DNA repair factors to come in, the chromatin environment must be prepared to receive these factors. We'll talk about that in a second. All right, so let's focus on the first one, which is the activation of the checkpoint pathways. So this is what we collectively call the DNA damage response, and it's not necessarily pertinent only to the repair of double-strand breaks or in response only to double-strand breaks, but to most types of DNA damage. But it becomes most relevant for the repair of double-strand breaks because, again, these are serious lesions and the cells need to make sure that they are corrected before cells go on into dividing. And so obviously in response to DNA damage, cells can either repair the damage first by activating checkpoints, as we will see in a second, promote DNA repair if the amount of damage is potentially repairable, or if the damage is extensive, the cells will either arrest permanently and exit the cell cycle through the program called senescence, or they die if the damage is so severe and simply cells cannot tolerate them. If the damage is not very severe, for example, like in the case of UV-inflicted damage, cells can start what we call tolerance program. And we've already talked about the translation DNA synthesis sort of quote-unquote repair program that is not necessarily able to repair the damage, but tolerate that damage so that the cells can maintain viability. All right, so how does the checkpoints get activated? It's get activated through the induction of checkpoint genes. And here, I think the cell cycle lecture becomes very relevant because all these different checkpoints are centered around halting the cell cycle progression by arresting cells at one of these three different major phases of the cell cycle. So there are three different checkpoints, if you will. One is a G1S checkpoint, which is an important checkpoint to prevent cells from entering S phase in the first place. So cells arrest in G1. The second one is cells arresting in S phase, and that's executed by checkpoint proteins that will arrest cells during S phase. And so that's called intra-S phase checkpoint. And then finally, and perhaps most importantly, and you need to think why, and come back in the question and answer session and tell me what your thoughts about that. The G2M checkpoint, where the cells arrest at the G2 or in mitosis in order to repair the damage if they happen to be encountering these double-strand breaks in that phase of the cell cycle, and they need to be arrested in that phase before they proceed on. All right, so the key players in executing these checkpoints are primarily two family member of kinases known as ATM and ATR. You probably heard about one or both of these guys. If not, this is a good time to emphasize their role. These are master kinases. These are the ones that initially sense double-strand breaks. ATM particularly is dedicated exclusively to the sensing and then propagating the signals through double-strand breaks. ATR is a sister homolog for ATM. Both are kinases. It has very similar homology, but they do have different set of substrates. And ATR, unlike ATM, responds primarily to single-strand breaks that happen during replication stress. For example, under conditions where the cells are treated with hydroxyurea or acetylcholine, the two agents that we talked about a little bit earlier. There's some overlap, and there's some evidence to suggest that both of these two proteins can actually compensate for the role of each other. All right, so what happens when ATM or ATR are activated in response to this signal? They activate different downstream effector proteins. And they do that by directly phosphorylating them. And two such proteins are CHEK2 and CHEK1. And from the name, you can tell that these are also kinases, that they are the mediators of the checkpoint activation. That's why they come with the name checkpoint. So CHEK1 and CHEK2 are checkpoint kinases that phosphorylate many downstream substrates that ultimately hold cell cycle progression. So here's a chart looking at cells going from G1 to S to G2M normally, but if they encounter damage, ATM and ATR get activated. They phosphorylate CHEK1 and CHEK2, and by doing so, they inhibit the CDC25 family of phosphatases that you heard about during the cell cycle lecture. And by doing that, what the end result is inhibition of the cyclin-dependent kinases, the main drivers of the cell cycle. And so by activating ATM and ATR, you inhibit cyclin-dependent kinases, and that's where cells are halted either in G1S, in S, or in G2M. Another important mediator of the checkpoint function is the very famous p53 protein. This is the most commonly mutated tumor suppressor protein in human cancer. More than 50% of all human malignancy have mutation in this important DNA damage response gene. And the most important function of p53 in response to DNA damage and stress also, because p53 has many more roles beyond its role in the DNA damage response, is to activate the downstream effector, most notably P21, which is an inhibitor for cyclin-dependent kinase 2. So P53 primarily induces G1S arrest by directly inhibiting CDK2 through the expression of P21. P53 is a transcription factor. It tetramerizes upon signaling from ATM as well as from CHEK2. So both of these kinases directly phosphorylate P53 at the N-terminal end of the protein. And when that happens, P53 becomes activated, it tetramerizes, and it goes to the promoter elements of P21 and transactivate P21 expression, which will then combine to CDK2 as well as 4 and 6, but this is a little bit less important, but CDK2 primarily. And when CDK2 is bound by P21, its activity is shut down and G1 to S transition is prevented. Of course, this is a simplified version of the schematic. You can see and appreciate from this diagram that there are many other layers of regulation that happen in response to DNA damage, particularly in response to double-strand breaks. There are many other kinases that get activated. DNA PK, for example, we'll talk about that in the context of double-strand break repair. But P53 also transactivate many different proteins in addition to P21 that we just talked about. And they're all sort of important for suppressing either the G1 to S. P53 also plays a role in the G2M transition. This is here only a sub-diagram that focuses exclusively on G2 to M. So P53 then is not only important for suppressing G1 to S, but also for suppressing G2 to M. So therefore, it's not surprising that when you lose P53, as in the case of human cancer, cells cannot respond very well to DNA damage. And that's why cancer cells in general, they are deficient in activating these checkpoints, at least a subset of them. Of course, I'm not expecting you to remember all these different factors. And so this is just to highlight the complexity of the pathway. All right, so let's focus on P53 because it's a really important mediator. As I mentioned, ATM and CHEK2, which are activated in response to DNA damage, they phosphorylate P53 as serine 15 and serine 20. These are N-terminal residues that get phosphorylated. When that happens, P53 can no longer be recognized by another protein called MDM2. So under normal conditions, when there's no DNA damage, MDM2 is an E3 ubiquitin ligase. I don't know if you still remember, I mentioned something about ubiquitination in the last lecture, where I mentioned that monoubiquitination of PCNA is important for activating the TLS program, the translation DNA synthesis program, by switching the different high fidelity polymerase from PCNA into the lower fidelity Y family polymerases that are able to accommodate lesions. So another form of ubiquitination, in this case, polyubiquitination, is a key step in ensuring that P53 is normally not expressed. So that's the function of MDM2. So MDM2 is a ubiquitin ligase. It's an enzyme that puts in polyubiquitin chain on P53, shunting it for degradation. In response to damage, however, P53 is phosphorylated at the N-terminus, both by ATM and by CHEK2. And MDM2 itself is also phosphorylated at a residue of 395. And both of these phosphorylation events disrupt this interaction. And by disrupting the interaction between P53 and MDM2, that allows P53 to accumulate in the cells and is able now to go and to bind to different transcription target genes, one of which is P21, and that drives the transcription of P21, which then goes and inhibits or bind to and inhibit cyclin E, CDK2, shutting down the S phase progression or preventing cells from getting into S phase in the first place. So that's the main function of P53 and its importance in the DNA damage response. So let's see what happens normally to cells. So here's a FACS diagram. Here on the y-axis, you're looking at total number of cells. So you've probably seen this before. So total number of cells versus DNA content. So this histogram typically represents cells that are in G1, cells that are in G2, the second lower peak, and anything in between are cells that are already undergoing DNA replication, cells that have intermediate contents of DNA between G1 content as well as and G2 content. So in response to radiation, for example, 7 gray here, you can notice and appreciate that all of the S-phase cell population has now been depleted, right? And cells are accumulating and arresting in G1. So these cells that normally will pass, these are asynchronous populations, some cells in G1, some cells in S, some cells in G2, and they keep cycling. These are normal dividing cell representations. So this is a snapshot of how they are represented. But when you expose them to 7-gray and you look 9 hours afterwards, you see that no longer you're able to see any S-phase cells, and the cells are arrested both in G1 and in G2. These are in wild type cells. In cells that are deficient of p53 or ATM, they're not able to respond to the DNA damage appropriately, and they'll lose the ability to arrest in S-phase. So you can see that the S-phase cells are still, you still have some cells that are in S-phase cells, as well as cells that are already in G1, they continue to progress into S-phase, and that's why this peak is lower than that peak. Now you notice that a lot of the cells still accumulate in G2, which means that the G2 arrest is still intact. So this is an important distinction between cells that are either proficient or deficient in some of the DNA damage repair pathways, is because cells, again, despite the fact that you're losing one or two of the checkpoints, in this case G1 and S, excuse me, you still have the ability to arrest them in G2M. And if you abrogate that pathway, cells will die. So let's see what the evidence for that. So here's a result from our own lab on glioblastoma cell line that are proficient for p53. They have intact form of p53. These cells were exposed to grays, and they were monitored as a function of time for their cell cycle distribution, depicted here as G1 population, S population, and G2 population. So let's see after exposure to 2 hours what happens. G1 cells are gradually increasing beginning by 8 hours. So you can see cells are pretty much arrested in G1. They accumulate in G1. Those cells that are in S phase, they'll continue going through S phase, but then they're becoming depleted because they're now entered G2 cells. So G2 cells are slightly affected. So this is depicted here by increasing in G1 DNA content. This is indicative of G1 growth arrest. Complete depletion of S phase as well as G2 cells. So the cells respond primarily by a G1 cell cycle growth arrest. Here's another glioblastoma cell line that is now mutant for p53. These are natural mutations that happens in some patients with brain tumors. And you can see here, and unlike the other cell line, these cells do not arrest in G1. They fail to arrest in G1. In fact, cells that are in G1, they continue entering S phase, and this is what you see, a little bit of bump here in the population of S phase cells. They're also not arrested in S phase. However, they are maintain their ability to stay in G2, pretty much the case. All right, so now that we have a good picture about what happens first is the cells arrest in response to double-strand breaks, primarily, now that we're focusing on this topic, through the induction of these checkpoint kinases downstream of ATM or ATR, and that results into cell cycle arrest either in G1 or S if they do have functional p53, or if not, then they'll arrest in G2 cells. The second thing that has to happen almost simultaneously is the recruitment of DNA repair proteins, and that depends on two main activities. The first is remodeling of the chromatin structure around the double-strand breaks, and the second is the loading of different repair proteins. So here's a snapshot about what happens very early on, immediately after double-strand breaks. ATM gets recruited. So ATM, as I mentioned, is a checkpoint kinase, but it's also a kinase involved directly in the repair of DNA damage through modulating chromatin structure. And so ATM goes and phosphorylate many substrates. One of these substrates is gamma H2X, or one of those histone protein, H2A. They phosphorylate that at a particular residue, serine 139 in the case of human histone. And that's important for further downstream signaling. ATM also recruit another complex called the MRN complex, stands for, it's a trimer of 3 different proteins called MRE11, in case you've heard it somewhere, RAD50 and Nibrin. These 3 different proteins come together, and what they do is these are what we call resection proteins, proteins that come and start resecting the double-strand breaks in preparation for their cleaning up. Of course, there are many other proteins that also come into play. One of them is a protein called RNF8, and one of them is called MGC1. Let's just focus on RNF8 for a second. This is an E3 ubiquitin ligase, meaning that is a protein that can put an ubiquitin chain on different proteins. One of them is BRCA1. This is the breast cancer susceptibility gene 1 that's mutated in many patients with familial history of breast cancer. And the other protein is 53pp1. It's much more complicated than that, but I just want to give you a snapshot that there are many different modifications that happen around the sites in order to prepare the sites for repair, even before repair begins. So one example is another modification on histones, where the cell sort of replaces the normal H2A histone with a different variant called H2AZ. And that's an important variant of histone that allow for the relaxation of the chromatin around the double-strand breaks. And that helps recruiting different factors again. The other function that's important along with this modification, this histone exchange process, is an acetylation reaction that happened at some of the histone gene tails. And that's mediated by a protein called TIP6. And histone acetyltransferases that add histones to particular sites. There are also a bunch of methyltransferases that will come and methylate some of these histone tails, and that's essential for the recruitment of 53pp1. Now, hopefully by now, you've read the paper that's assigned for today, the shielding paper. And as you hopefully have read it, 53pp1 is a really important protein for making the decision on what particular repair pathway has to be executed, depending on where in the cell cycle these cells are when the double-strand breaks are encountered, as well as on the availability of different repair factors. Generally, there are two main repair pathways the cells can repair these double-strand break with. One is homologous recombination repair, and one is non-homologous end joining. Of course, by definition, homologous recombination repair depends on the availability of a sister chromatid in order to take that sequence and repair the damage at this particular strand. So this, by definition, then, has to happen only when the cells already had undergone DNA replication, when you already have an intact sister. So this process is a very high fidelity process, meaning that the cells will simply take the information from the sister chromatid and use that to complement the severed strand or the severed double helix on the sister. And therefore, homologous recombination repair is only active in G2 or maybe even sometimes late S phase. Of course, if you're not in this phase of the cell cycle, you don't have that privilege. You don't have a sister from which you can copy the genomic sequences, and you're left with a break, and the cells have no idea how to repair that break. So therefore, it's been very long thought that the process of repairing this lesion is kind of error prone, but more and more evidence suggests actually that it's not necessarily error prone, and I'll show you more examples of that. So non-homologous end joining, therefore, the name comes from the fact that this repair process is not dependent on homologous sequences, and that's why they call it non-homologous. And end joining, meaning simply by joining the two ends of the double-strand breaks. So we have two programs, non-homologous end joining program, where you don't have sister for homologous recombination to happen, or homologous recombination when you have something from which you can copy genomic DNA sequence. And clearly now that we said the cell cycle position, the position of the cells within the cell cycle is a major determinant for which of these two pathways will be utilized. So, so let's go over these programs. Again, in today's paper, we'll deal about the shielding complex. And as you notice, this is a recent paper. So the field is still evolving, and we're still learning some of the factors that are important for making that decision. So the decision is primarily dictated by this protein called 53pp1. In its presence, it's able to recruit the shielding complex that the paper deals with, and hopefully that will trigger some stimulation during the discussion session. And what that does is that it binds to the single-stranded overhangs that come from these double-strand breaks and prevent access to extranucleases. And therefore, what they do confer to the cells is what we call end protection. So in the presence of 53pp1 and downstream factors, right, you don't have the ability to excessively resect the double-strand break ends. And when you do have that pathway intact, and in the absence of homologous recombination repair, cells repair the damage through the canonical pathway of non-homologous end joining. This is dependent on the presence of a heterodimeric protein called Ku, or Ku70 and Ku80. These two guys respond by binding to the ends directly, they then recruit DNA protein kinase C. This is a family member of ATM. Remember, ATM is also important for recruiting 53pp1. So DNAPK is a family member of ATM, but it has different set of substrates. And so DNAPK then forms a trimer with the Q70, Q80, forming what we call the DNAPK holoenzyme. So DNAPK catalytic subunit alone is not active, but when it's bound to the Q70, Q80 at double-strand breaks, it becomes activated, and they form this holoenzyme trimeric complex. Then additional protein come into play, including some polymerases, polymerase beta, beta, eta, or lambda. And these polymerases will fill in the gap if there's any gap that's needed. And obviously these are TLS polymerases, meaning that they can simply fill the gap with some nucleotide after cleaning the ends. And then DNA ligase 4, unlike 1 and 3 that we talked about in the last lecture, ligase 4 would specifically come in and ligate the two ends together. If there is no information loss at these breaks and you have simple, readily ligatable ends, you don't need anything more than these 3 different proteins. But if you do have some problem with the ends, then the cells have to find a way to repair these ends using some additional accessory proteins, and that will result ultimately into some form of errors. But if you do have simple, clean cut in the genomic DNA, this process of repairing the damage is very high fidelity, and I'll show you evidence for that in a second. Of course, this all have to happen in the presence of 53pp1 and in the presence of intact signaling to the shielding complex that prevents extensive resection. So what happened if you then, so let's back up a second, and so let's just go over this one more time. So when you have double-strand breaks, you could have the normal recruitment of 53pp1 along with the shielding complex. You sense the double-strand breaks by the ATM and the MRE complex. Then the ends are readily recognizable by Q70 and 80. They recruit the DNAPK catalytic subunit. Together, they form the holoenzyme. If some cleaning is needed, then another protein called Artemis will come in and do that job. You'll fill in the lesion with DNA synthesis polymerases. 3 of them have been implicated in this process, and DNA ligase 4 will come in and repair the damage. And this is typically an accurate form of repair. This is just a depiction of that outline right here. So ATM signals, mRNA complex come in, they signal the recruitment of Q70-80, they bring in the catalytic subunit, together they form the holoenzyme, processivity by Artemis, polymerization, and ligation. Okay? If you don't have an intact Q70, Q80 signaling pathway, then you'll end up with repairing the damage through a different pathway. And that's what we call the alternative non-homologous end joining pathway. The alternative non-homologous end joining pathway is a version of non-homologous end joining pathway that depends on ligating the ends after some limited end resection, some limited form of end resection, maybe few nucleotide on both ends. And then the ligation happens using some form of microhomologies that are flanking the double-strand breaks. And that depends on the activity of PARP1 and XRCC1. PARP1 is the same protein that we've talked about in the context of nucleotide excision repair, in the context of base excision repair. So if you don't have PARP1, or if you have PARP1 inhibitors that we talked about last time, not only that you inactivate the base excision repair, but you also inactivate the alternative branch of non-homologous end joining, and therefore you're depriving the cells of 2 different repair pathways. In addition to these 2 proteins, you also need polymerase theta, as well as DNA ligase 3. And that form of alternative non-homologous end joining, because of the microhomology that's present here, typically results into small deletions and insertions. They're collectively called as indels. You'll hear that term over and over. So N stands for insertions and del stands for deletions. So small indels, typically less than 20, 25 nucleotide. So whenever you see double-strand breaks that are being repaired with the loss of these small sequences or the gain of small sequences, that's a signature for the alternative non-homologous end joining program. In the absence of either of these, then there's a third program. There's a little bit different version of it that depends on the presence of XPF and ERCC1. If you remember, XPF and ERCC1 are part of these nucleotide excision repair that functions during the XP-dependent pathway, the nucleotide excision repair, the genome global pathway. Of course, it utilizes another protein called RAD52, and together they sort of allow for the two ends to come together. But now the ligation depends on the presence of longer microhomology, and typically when repair happens through this mechanism, it results into formation of large deletions. And so this process is called single-strand annealing process because they depend on the annealing, long annealing between single strands at the double-strand break sites. All this happens now when the cells are not in G2M. If the cells are in G2M, then you have the ability to proceed repairing the double-strand breaks through the homologous recombination pathway. And that requires, number one, that you inhibit shielding so that the ends of the double-strand breaks can be resected to a long sequence, so you have longer overhangs. And that requires activity of two enzymes. One is the plume helicase, and obviously helicase because you need to unwind the DNA helix around the double-strand breaks, and you need an extra nuclease, and a nuclease that will come in and chew these overhangs so that can form a longer stretches of single-stranded DNAs. Because they're single-stranded DNAs, they need to be stabilized. For that to happen, you need a protein called RPA. This is an RNA binding protein that gets later on replaced by another protein that binds to single-stranded DNA called RAD51. BRCA1 and BRCA2, or BRCA1 and 2, are important players in promoting this homologous recombination repair by promoting what we call the strand invasion, where one long overhang will come and invade the sister homolog, or if you have the homologous sequence in the vicinity, then that single-stranded overhangs will come and invade that sequence, and it will copy that information and allowing for, after the step of resolution, allowing for the sequence to be generated using that sister strand. So here is a depiction of that phenomena. Again, ATM and ATR are important in this, in signaling the initial events to allow for chromatin remodeling, recruitment of the MRE11 or the MRE complex, sorry, MRN complex that's made of these three different proteins. Resection begins with this trimer, but is continued by the extranuclease 1 that we talked about right here. And so you'll form longer overhangs. They are stabilized by RNA binding protein RPA1 and other versions. And then that gets replaced by RAD51 and RAD52, which stabilizes the end of these long single-stranded DNA. Then strand invasion takes place through the help of BRCA1 and BRCA2, and then resolution and crossing over between the two sisters is happening, and you end up with perfectly accurate repair. So homologous recombination repair, or homology-directed repair, sometimes it's called, so HR or HDR, is an important step for maintaining the fidelity of DNA. If you inactivate BRCA1 or BRCA2, as in the case of breast cancers, cells are not able to repair damage through this high fidelity pathway, and they're dependent completely on the non-homologous end joining pathway or on single-strand annealing that we just talked about. So let's put it all together. Okay, so when you have a double-strand break in the cell, cells respond by activating the DNA damage response. This activates checkpoints and slows down or prevents the cells from continuous cycling. At the same time, they remodel the chromatin in the vicinity of double-strand breaks, and then 53BB1 is recruited through ATM and many other proteins and many other modifications at the chromatin. That recruits shielding to prevent hyperresection, and if that happens, cells typically repair their double-strand breaks through the Q70, Q80 dependent canonical non-homologous end joining pathway. If shielding is defective or 53BB1 is not recruited quickly enough, then cells have the ability to use a backup pathway, the alternative non-homologous pathway, which is PARP dependent and utilizes both theta and DNA ligase 3, and that typically comes in with small indels. If that's also absent, cells uses single-strand annealing process to generate larger deletions and more mutations. Of course, in the presence of homologous template, cells inactivate shielding, and hyperresection takes place, is initiated by the MRN and another protein called CTIP. You don't have to worry too much about the name, but these will initiate the resection, that will then be extended through the function of exonuclease 1 with the help of helicase, Bloom helicase, and will start forming these long overhangs that will be stabilized by RNA binding proteins, RPA and RAD51. BRCA1 and BRCA2 will help the strand invasion process, and then resolution happen, and you'll end up with high fidelity repair. Okay, so hopefully you have a good picture on that. So we'll keep that picture during the Q&A sessions to help us guide the discussion. And please think about questions here so that when we come to the discussion, we have some interesting insights of what would happen to the cells under different conditions. All right, so where did the problem then come from when we have these double-strand breaks in the cells? Well, they come depending on where the cells are. If they are in a pre-replicative situation, for example, when the cells only have one sister of the two chromosomes, then you could have breaks happening anywhere. And if that's happened, for example, at this arm and at this arm of two different chromosomes, maybe they're nearby, you have the potential for ligating the different ends, unless you have a way to stabilize these ends together. And there are proteins that are known to be involved in that. And when they're disrupted, you then have no way of stabilizing the ends, and you could have the shuffling between different segments. And that's a random process, depending on how many breaks there are. Excuse me. Then if that happens, you have the potential of, for example, here, having one segment. This is missing another centrosome here. I'm sorry. So this should be here also present. So you'll end up with one long chromosome that has two centromeres, like here. So when you replicate this segment, it'll end up with something like this. And obviously, this one has two centromeres, which mean that when they start to align during metaphase, as you've heard from Todd 2 days ago, you'll have the inability of the cells to recruit this particular abnormal chromosome to one particular pole of the cell cycle, because each pole may have the chance to pull that chromosome to its direction. So you could have one spindle pulling on this side and one spindle pulling on that side. And if that happened, then you have two poles competing for the same chromosome, and then that will not segregate well. And ultimately, that would result either in their breakage or in one cell getting both of these structures and one cell get none. The other segment, of course, doesn't have any centromere. And so this segment, or what we call acentric fragment, because they don't have a centromere, they'll ultimately be shuffled between the two daughter cells randomly. And so one cell will get it, the other cell may not get it, one cell may get both copies, or two cells may get one copy, so depending on what's really happening. Of course, this is very simplistic if you only have two single double-strand breaks, right? Of course, under a condition, for example, with radiation, let's say 2 grays, which is typically done in fractionated therapy today, every single cell will encounter about 40, at least 40 to 50 double-strand breaks. So you can imagine the amount of shuffling that could happen in the cells. And these are what typically causes lethality when the cells undergo mitosis. They are not able to segregate their chromosome normally, and they'll die by what we call mitotic catastrophe. Also in the G1, you could potentially have 2 breaks on 2 different arms of the chromosome. And if that happens and you ligate these arms, you'll end up with a circular form, or what we call incorrect union between the 2 arms of the same chromosome. This other fragment will also be an acentric fragment as well, and that will be randomly distributed. And when this one replicates in S phase, then it will give you this overlapping rings, which you can detect by analysis under the microscope, looking at metaphase spreads. Now, the situation is quite different if the cells already have undergone DNA replication and you have 2 sisters linked together through their centromeres. And here, you could potentially have double-strand breaks that will be encountering the 2 sisters, where now the ligation happened between the 2 sisters, and so you end up with a sister union. Of course, this also causes problem because they're not able to segregate normally during mitosis, and that will result into the formation of both of these structures, dicentric chromosome as well as acentric chromatin fragments, this guy right here. So many different aberrant joining that will happen. And all of these result from non-homologous end joining if the program is not able to correctly ligate the correct ends together. Of course, with multiple double-strand breaks happening at the same time, this is highly unlikely. And that's why double-strand breaks are very lethal. You could have a non-lethal double-strand breaks, as we mentioned, if you have symmetric translocation between the different cells, where the joining just happened to be between two exchanging chromosome sections. So here and here, and simply this section that belonged to this chromosome now is part of this chromosome and vice versa. So this is a normal translocation event, but that's important for cancer etiology, actually. Many cancers have these translocation events. And some of these translocation events are not very random. They're actually very selective, meaning that cells that have a particular translocation, for example, here, BCR-ABL, this is translocation that made some abnormal junction between two different genes that now have a new function altogether. That's very common observation in chronic myeloid leukemia, for example. There are many other common translocation events. For example, the MYC IgG. This is where you have a break that will bring in the MYC gene, which is an oncogene, meaning it's a gene that drives cell cycle progression forward under the control of an immunoglobulin heavy chain, which is normally expressed only in B cells and drives very high level of transcription. So when you put in an oncogene under the expression of a high promoter, right, you end up with expressing many, many, many copies of MYC, and you end up with an oncogenic transformation. This is a typical signature for Burkitt's lymphoma. Many other examples, including prostate cancer and the Tempus erga. So symmetric translocation, even though they're not necessarily toxic, they could have some serious consequences and potentially contribute to cancer development. Okay, so now that we've gone over double-strand breaks, we'll just dedicate a moment on the repair of interstrand cross-links. Sorry, interstrand cross-links. These are cross-links between the two strands of the helix. Obviously, this causes problem because polymerases cannot go through. Transcription also would not be able to pass beyond that, because for these two processes to happen, you must unwind the DNA. You must use only a single strand to allow for the transcription or for replication to take place, as you've learned in the case of replication, and you learn in the case of transcription. These lesions also impede the recruitment of many different factors, like transcription factors, normally helicases and nucleosomal structures. So they have some serious consequences, and they must be repaired altogether. So how do cells repair them? Well, number one, they repair them by activating the DNA damage repair pathway, right? The DDR. And so let's see how the process of preparing these lesions takes place. So when you have a lesion like this and you have an ongoing replication fork, this is a lesion, for example, that can be used, generated by platinum compound. These are commonly used in cancer therapy. So here is what happened to patients or to cells from patients receiving these therapies. These cancer cells typically are heavily dividing, meaning that they're always replicating their DNA. And so when you have a replication fork going through, and you'll see this lesion, obviously the polymerase stalls. And when that stalling happens, a whole bunch of proteins come in the vicinity and they get recruited. These set of proteins called collectively the Fanconi complex. You probably heard the name Fanconi anemia. This is a typical disease that's characterized by an anemia and also by extreme sensitivity to DNA cross-linking agents. And from the name, you can imagine that these genes are all involved in the repair of these damages. And so you have about 18 or so different protein subunits together. They come together to recognize this lesion. And mutation in any one of them will cause Fanconi anemia, will cause this problem. By the way, Fanconi D2 and Fanc-I, these are the BRCA1 and BRCA2 that we've heard about in homologous recombination repair. But the rest of the complex is all needed for this recognition of this abnormal structure. So when they're recognized by the Fanconi complex, then one protein complex is recruited, and that's the XPF-ERCC1. Now remember, this is the same two proteins that are important for repairing nucleotide damage that are repaired through the nucleotide excision repair pathway that we talked about. XP stands for xeroderma pigmentosa. F is group F that was discovered through some genetic mutations. And ERCC1 is an accessory factor. These two together, they represent a structural specific nuclease. These are the ones that are able to induce dual incision around the structure. So they specifically recognize the structure. They make far away incision on one of the strands across the lesion. Okay. After that, an unhooking process has to happen. So now you release this segment from this fork. And so you're having this is intact, but now you have a double strand breaks right here. Right? You also have now a single strand break with this structure that is now flipped to the outside. This is called unhooking process, flipping it to the outside. So let's see how the cells then recognize the structure. All right, so a protein called SNMA1 will come in and will start digesting this little alkylating adduct, basically the two nitrogenous bases that were covalently linked together, and that will be digested by the SNMA1, leaving in the process a single-stranded break on this fork and a double-strand break on the other fork. Now, of course, a single-stranded break has to be repaired, and that's repaired through translation DNA synthesis. Well, before that to happen, cells uses this strand, the damaged strand, to form, to replicate the sister for that strand right here. And so this replication then has to happen against a bulky lesion. And to allow that to happen, you need the TLS program, the translation DNA synthesis polymerases. And in this case, it's one of them. It's called polymerase zeta. Along, of course, with the ligase one that will ligate these ends together. And so now you use the damaged strand to fill in the gap on the sister of the first replication fork strand. The double strand breaks on the second one is still intact. So you still have a double strand break, and you still have this digested element by the SNMA1. So now you need the same dimer again, the XPF ERCC1. So again, a nerve pathway will come in and make now two incisions around this strand. So in the previous picture, I showed you that the same enzyme makes two incisions across one of the two strands that are opened up to allow replication of one of them to take place. In the second step, after you already filled that gap right here on this strand, you still have a gap on the second strand. We'll deal with this later. But for this one, then has to be removed by the same structure-specific endonucleases. And again, now this one will use this information from the newly synthesized strand to restore that sequence. So that's another nucleotide excision repair step. So we began with a nucleotide excision repair step, with a TLS step, back with NER step, and you still have a double-strand break that must now be repaired. But now this looks very similar, like a normal double-strand break, right? So how does that get repaired? BRCA1, BRCA2, the same factors that we talked about early on, the same homologous recombination pathway. So this is why homologous recombination pathway is the final and most important step in repairing these lesions. Let's recap for a second. So you have a lesion, a fixture between the two different helixes on an ongoing replication fork on both sides. One is a leading strand and one is a lagging strand. A nerve factor will come in and make a dual incision on one of the strands, flipping it outside by unhooking mechanism. This requires, of course, recognition of the lesion to begin with the Fanconi complex. Removing of that by an SNMA1 digestion process. TLS will come in and fill the gap using the information on this strand. Okay. NER will come again and make the dual incision, removing this altogether and using the newly synthesized strand of that sister to restore the sequence. Obviously, because this is potentially also error prone, this could have a mutation here in the vicinity of this sister. You're still left with a double strand break that gets repaired through homologous recombination repair that we just talked about. So here's just a summary of some of the diseases that are seen upon deficiency in one or more of the different DNA damage repair pathway, and boxed here in red are those that are involved in double-strand break repair, most commonly the ATM. ATM, by the way, stands for ataxia telangiectasia. From the name, you can see these are patients that have spider veins, similar to what we see with the XP. But also they have ataxia, a very specialized form of ataxia. This is an abnormal movement, mobility of these patients. They're very sensitive to these patients. They have increased frequency of different cancers. They have extreme sensitivity to gamma ray, and they obviously have deficiency in ATM kinase, and they're not able to repair. First, they're not able to sense to the DNA damage repair pathway, and second, they're not able to participate in the repair process. BRCA1 and BRCA2, as I mentioned, these are two important proteins play a role in homologous recombination repair, and they are frequently lost in patients with familial breast cancer. Also, these patients tend to develop ovarian cancer at a very high rate, and these cells taken from these patients are deficient in homologous recombination repair. Some additional factors like Fanconi anemia, for example, these also important for homologous recombination repair, but particularly in the context of interstrand cross-links. And that's why patients with these, with mutation in any of these genes, many number of genes, causes Fanconi anemia, have heavily accumulated genomic instability, and they have extreme sensitivity to cross-linking repair. There are additional lesions as well that are not in this table, but I'm not going to talk about that. Okay, so just in the last 5 to 10 minutes or so, I'll talk about what else can cause double-strand breaks, and can we use that in research? And the answer is yes. For a very long time now, people have used many different agents to induce double-strand breaks artificially. And one of them is through the use of restriction endonucleases that you've heard about in many of the undergrad studies. These are nucleases that are typically large in size. Typically, they came and discovered in bacteria. And so they don't have mammalian homologs, for example. These are bacterial-specific restriction endonucleases. They're actually evolved to recognize foreign DNA viruses. So when you have viruses infecting bacteria, bacteria have these restriction endonucleases that would recognize the sequences that are not present in the bacteria, but present in any foreign DNA. And so this is an immune mechanism for bacterial cells. But people have used these restriction endonucleases to express them in mammalian cells to induce specific cuts somewhere in the genome. And you can imagine that you could put in some sequence in the genome through some many different technologies now, where you know where exactly you put in that sequence, and then you can now express the relevant restriction endonuclease to create a specific cut in the cells, and ask the cells, how does the cell repair it? One example of that meganuclease is a protein called ISCE1, which you probably heard of in the shielding paper that you discussed. So this is a large restriction endonuclease that recognizes about 18 base pair site. Other more newer versions of protein that have the capacity to induce cuts in the genome in a site-specific fashion, and they've been used very heavily now in generating transgenic mice, where you can manipulate the genome of mice in vivo. And these are depending on zinc finger nucleases, or what we call talons. These are also largely modified protein structures, usually in multiple subunits, but they do have one of the domains that have the ability to cleave DNA specifically at certain sequences. These are much more elaborate. They take a lot more effort to design and to generate, to be able to generate double-strand breaks at a sequence specific fashion. Of course, with the discovery of CRISPR-Cas9 that you must all know about and heard about by now, excuse me, it became so much easier to actually design a way to induce cleavage at many different sites in the genome in a sequence-specific fashion that can then be repaired either by non-homologous end joining or by homologous recombination repair, as we discussed. See, this graph actually was taken from my colleague here, Mazhar Adli, who has just left us last year and joined a different faculty department in Chicago. But he was an expert in the CRISPR-Cas technology, and this is one of his figures that taken from one of his reviews. So CRISPR-Cas9 actually is a very elegant way, and again, this was discovered in other organisms. Now we have many different Cas nucleases. The numbers keep increasing from all kind of different form of lives. They're obviously not present in mammals. These again are also part of the immune system of many different organisms against invading viruses. And what these enzymes do is that they depend on recognizing foreign DNA sequences through annealing with an RNA primer that will come in and anneal specifically with a particular sequence. Most importantly, what we call the PAM sequence, or proto-excision motif. This is an important sort of guide to where the Cas9 will come into the genome and induce a specific cut using its nuclease domain. So by simply expressing then Cas9 and any given sequence that anneals to any part of the human genome by 20 different nucleotides, you can actually engineer a way to induce a specific cutting anywhere in the genome. And that has revolutionized how we actually do genomic editing in cells. And not only that, but also now people are using it for gene therapy in order to correct or delete certain genes from a whole animal. And it's been very successful. In my own lab, we actually used Cas9 guide RNA or small guide RNA Cas9 to generate 5 different transgenic mouse models where we induce specific cleavage in the genome of these animals globally, so that we can actually inactivate one gene entirely from each of the cells in this animal. So this is a very powerful system that you'll hear about more and more, and you'll get to learn and love at some point. And I think there'll be some lecture dedicated to that at some point during the course. I'll just talk briefly about how to actually measure double-strand break. So to measure double-strand break repair activity in cells, people have used different ways of doing that. So the simplest way was used very early on was to use a plasmid that has been linearized in vitro. You cut it into half, and sometimes you could actually have, for example, a plasmid that you cut it in half so that one end has a GFP fluorescent coding sequence and a promoter, and you linearize it so that GFP will never be expressed until you actually ligate these two ends together. To ligate them together, then you need a repair system that will bring these two ends together and allow for the expression of GFP. So people are be able to then measure repair by simply measuring how many of the cells will then express an intact green fluorescent protein or any other fluorescent marker for that reason. You could also put in an antibiotic marker also in a linearized plasmid and ask the cells, how often do the cells fuse the segments together to produce a productive antibiotic resistant gene and select for genes that will select for cells that will survive the antibiotic selection. And that also will give you a measure for how much repair the cells is able to do. More recently, people have started using fluorescent reporters. This is where you put in a reporter. For example, here, you could put in a reporter that's made of subsegments of a GFP sequence, so this G and FP. If these two together come together in some fashion, then they will fluoresce green. But in that reporter, what they do is they put in some foreign sequences, and they have, in this case, 2 restriction enzyme site recognition for that ISE1 restriction enzyme that I told you about. So if you put in this interchromosomally, you can actually integrate this in the chromosomes, and then you could transfect cells with the ISE1. What ISE1 will do will come in and will induce a cleavage here and here, remove this segment and allow for the GFP to come together as in here and allowing for the expression of GFP. So by simply expressing then ISE1 enzyme into the cells, you can ask how frequent these cells repair the GFP construct and allow for GFP expression or the expression of any RFP. In my own lab, we actually developed a new system. This is a paper that hopefully will be published very soon, where we tried to overcome some of the limitations in this reporter assay system. So this was dependent on, again, Cas9. This is a lentiviral construct that we call it CDDR, which stands for lenti-CDDR, stands for CRISPR-Cas9 based dual fluorescent double-strand break repair reporter. So the CDDR reporter then has expresses mCherry from a strong promoter, but flanking the mCherry is 2 halves of the GFP. And then flanking the mCherry, you have 2 important sites that we generated or engineered that can be recognized by a small guide RNA if you also express Cas9 endonuclease into these cells. So you can make a stable cell line with this construct by infecting them with the virus expressing this construct. You can select the cells by plasticity because this construct has plasticity resistant marker, and all these cells will be lighting up red, right? Of course, they're not going to be lighting up green because green is not a contiguous segment. If you then express Cas9 along with a guide RNA that targets this site alone, then depends on how this is ligate this end, you will either get cells that are mCherry positive cell or that they have lost mCherry positivity altogether. If you express Cas9 along with 2 different guide RNAs, A and B together, on the other hand, what you'll end up with is breaks here and here. And if this segment is lost, the GFP halves will come together and the cells will light up green and will be no longer red. So green and no longer red will indicate high fidelity repair between these 2 ends, whereas in the context of single cut, the loss of mCherry will indicate mutagenic repair, or repair that's dependent on the non-homologous end joining branch that results into indels, deletions or insertions. So let's see how this works. So again, normal intact construct into the cells. You expressing Cas9 along with guide RNA that target this site. You either get this or that, right? Either cells remain red or you lose them. And that's exactly what we see. So here cells normally do not fluoresce any M-Sherry. So these are M-Sherry negative cells. If you put in the construct, all the cells are red. If you transfect these cells now with the guide RNA along with Cas9 that target the first site, then the bulk of the cells shift back to becoming non-red again. And you can quantitate that in different clones. So this is how much mutagenic repair the cells can undergo. About 80% of the cells are undergoing that. You can then also PCR the junctions from these surviving cells, and you can send that for deep sequencing after you make library. You can trim the adapters. This is a complicated process. I'm not going to get into it, but the point is you can actually sequence this junction from a pool of cells. And you can then ask the question, what fraction of these sequences have deletions? What fractions have insertions? You can even look at what's the sizes of these deletions. So here in 4 different clones, the bulk of the deletions are about 20 or to 30 nucleotide, and then much less a number that are greater than 30 nucleotide long. You can also see how many insertions at these, how many sites, right? So remember, because each cell has one copy, so each read basically represents one cell. So you can see here that 80% of the cells will have one nucleotide insertion, and only 10% or so 2 nucleotide, and only less than 5% of the cells will contain less than 3 base pair insertions. So you can actually look at the distribution of the different repair outcome that come from expressing Cas9 and guide RNA to induce single cuts within intact cells. All right, so what happens if you induce the cutting at both of these ends? You'll end up with cells that are, some are red, some are green, right, like here, and some that are neither red nor green. So neither red nor green, these are cells that repair the junctions. They survived, obviously, because we can amplify them and collect them, but they have not been able to restore M-Sherry, and they're not able to fuse the 2 GFP together. So these cells undergo mutagenic repair. Basically, repair that does not fully restore the junctions. Or cells that are fully restore the junction at each of these 2 ends, and these will remain red, or more importantly, cells that will bring in these 2 faraway ends together in a perfect ligation reaction. Cas9, by the way, induces cuts at a very specific sites within that sequence, so we know exactly where the cuts are happening. So then if they're repaired correctly, we know that we're perfectly at a single base resolution fuse that repair perfectly. And that's why these cells light up green. Here's an example. If you look at that by FACS, so here we're looking at GFP on the y-axis, M-Sherry on the x-axis. Normally cells are all red, but after inducing double or 2 double-strand breaks in that construct, the bulk of the cells will turn green, or some cells will turn green, about 25% of them. Some cells will remain will remain red, and the bulk of the cells will lose both expression. So we can also do PCR across from here to here. And if you do that, normally you only have 1 band. But if you now use 2 cuts and do from here to here, you have 2 situations. Either this band come together and this band come together, you still maintain the same parental band, or you lose this altogether and you bring in these 2 together, and now you lost this 700 base pair. You're coming up with a much smaller PCR fragment. You can then take this fragment and PCR sequence by high throughput sequencing and do the same thing that we talked about before, and measure how much of this that resulted into the fusion of the 2 faraway ends is actually indel free. And the result is outstanding. About 70% of all sequences do not have any mutation. They perfectly ligate these ends. And these are what's scored as green cells. Right? The remaining cells are mostly having deletions, some insertions, and some base pair substitutions. And you can look at the details of these all together. You can also supplement, along with the Cas9 and the guide RNA, another plasmid that doesn't have a promoter, but instead of the M-Sherry, actually has a blue fluorescent protein. And if you flank that with 2 homology arms to these GFP, this then functions as a sister to allow for homologous recombination repair. And if you induce a cut here, and you have in the cells a sister that has homology here and here, this BFB can potentially be recombined instead of the M-Sherry, and the cells will light up blue, right? Only if the BFB comes exactly in the right position here under the same promoter. And that would be indicative of homologous recombination repair. So you can actually quantitate homologous recombination repair if you only add one additional plasmid. So this scenario then will encompasses the induction of a single double-strand breaks in the cell that are transfected with a donor plasmid. And you can ask the question, how many of them will repair by non-homologous end joining here, or by alternative backup non-homologous end joining mutagenic pathway, or by, or what did I have here? Or by homologous recombination repair, and you can quantitate that these cells will light up blue, but will be negative for M-Sherry. Obviously, there's no green here because there's no induction of double-strand breaks that will bring these two guys together. And that's what you see on effects analysis by looking at BFP gating. You see homologous recombination repair events are much less frequent than any other type. Then you can actually ask the question, what happened if you delete one particular repair factor? And so here's an example when we deleted the 3 different proteins involved in the canonical pathway of non-homologous end joining that we just discussed, the Q70 dependent pathway, right? So ligase 4, XRCC4, and XLF. So we generated one single parental cell line that has the CDDR reporter, and then we deleted ligase 4 in 2 different clones, or XRCC4 in 2 different clones, or XLF in 3 different clones. One of them has some residual expression, so we exclude this one from the analysis. And if you do a single cut, you'll see that the loss of these genes increase the mutagenic repair. In other words, when you lose these factors, the cells are dependent entirely on the alternative branch of canonical pathway. And you can look at their distribution of the different deletions. So when you do that, in the cells that are deleted of these canonical non-homologous end joining factors, the bulk of the cells now have deletions rather than the normal distribution between deletion and insertions. You also notice that the deletion sizes are becoming larger. That's signature of the alternative branch non-homologous end joining pathway. In other words, you deprive the cells from the canonical pathway and you became completely dependent on repairing them through the alternative branch, which has typically larger deletions and larger insertions. You see the power of the system now. So what happened if you do double-strand breaks, 2 double-strand breaks in that construct that allow for the generation of blue cells? Well, number one, you lose the ability to generate any green cells. So this is, you shut down the ability to ligate the ends with any fidelity. So you're no longer able to faithfully repair the cuts. Cells are still surviving because they repaired the cuts, but through entirely a mutagenic pathway, as you can see here, by the loss of green and the loss of red, right here and here, and all the cells now become non-fluorescent. This is just the same thing that we've looked at before, but now in the context of double-strand breaks. Again, instead of the very high fidelity repair, now all the cells have large deletions here, right? Mostly larger than 30 nucleotide compared to the control cells, many more deletion than in the control cells, and a lot less insertions that are typically larger in size. You can also look at these microhomology signature I talked to you about. In the case of the alternative non-homologous end joining pathway, they often utilize microhomologies. So when you look at the reads that come from the high throughput sequencing, you'll see that this sequence right here, GGA, for example, and this GGA right here, you see there's a microhomology here, triplet and a triplet. And what you end up with is the deletion of these sequences that are immediately flanking the homology sequences, resulting in the removal of one homology arm. You see this here, you see this here, you see this here, in every area that you see the box. So we can quantitate in these reads how many of the different reads in the cells that are deficient of these proteins are now having these microhomology signature. In normal cells, it's about 50%. In cells that are deficient of these canonical non-homologous end joining branch, they're now over 80% of them. They have these microhomology signature. You're enriching for repair through this other pathway. So now you have a competing pathway is taking over. You also have tendency to increase the ability of the cells to actually repair by homologous recombination repair. If you inactivate the canonical pathway, you have higher tendency to repair through HR pathway. Of course, if you do the opposite by removing some HR repair pathways, then you're decreasing the ability to repair through the canonical non-homologous end joining pathway. So these pathways are competing with each other, as you can see here from this. This is just another version of the diagram that I showed you that outlines the 3 different or the 4 different repair pathway: canonical pathway, alternative pathway, the single-strand annealing pathway, and the homologous recombination pathway. So if you shut down the expression of any of these proteins, as I just showed you in our own example, you're boosting HDR because you're no longer dependent on this pathway. You cannot utilize this pathway because it's intact. And you're having much larger deletions that's dependent on single-strand annealing, more so of alternative non-homologous end joining with a microhomologous signature, and larger deletions and larger insertions. And also you have the ability to repair the breaks through homologous recombination repair with higher frequency. Okay, so hopefully from that example, I showed you that it's really fascinating how the cells devise many different competing pathways to repair these lesions. Cells cannot leave these lesions unrepaired, and there are many different efforts and a backup pathways, right? The canonical pathway and the backup pathway that's normally not active as a way to ensure that these double-strand breaks are repaired. So that ends the second lecture of the DNA damage repair, and hopefully that was something that was joyful to listen to, and I'm looking forward to your question during the Q&A session. See you then. Goodbye.